Okay, in this chapter, we're going to talk about cell structures, but first, we're going to talk about the origin of the first cells, because when the Earth first formed, it was lifeless. And then at some point, there were cells. So let's talk about what scientists think happened to get to that point. And this directly addresses cell theory, which we mentioned at the beginning of the semester. So cell theory is, and remember, a theory in science means that it's backed up by lots of evidence and it's generally accepted. So all organisms are composed of cells. So everything that we think of as alive today has at least one cell. And the cell is the smallest thing that we consider alive right now. So if we break cells apart into their components and we pull out the chromosomes or some part of the cellular machinery, like the ribosomes, we no longer consider those individual bits to be alive. And right now, cells only come from the division of other cells. But at some point, that was not the case, because right now, new cells are not appearing spontaneously. But there had to have been a period in the early development of life on Earth where there were things that weren't cells, and then there were some sort of protocells that were cell-like, and then there were the actual first cells. So let's talk about what that means. This portion of science, the science of the origin of life, is one area where we just don't have a lot of data. And all we have are hypotheses. We don't have a theory about the origin of life because, of course, a theory requires lots of data and it has to be generally accepted. So I'll talk about three of the most popular among scientists hypotheses at this time. But I'm going to deliberately use the word hypothesis for each of these ideas and not theory, because we simply don't have enough data to have a theory about the origin of life. And we may never know, because the origin of life was simply too long ago, around 4 billion years ago, and we simply may never have enough data to know for sure. But that's okay. Scientists like hand-waving and speculation and arguing as well. What we do know is that of the cells dividing on Earth today, all of them are related by descent. So, and we certainly haven't studied every form of life on Earth. There are hundreds of thousands of species of single-celled organisms that haven't been studied in detail. So it's possible we could discover some weird bacteria-like thing somewhere that's not related to the other cells on Earth, but that hasn't happened yet. Of the thousands and thousands of species that have been studied using their DNA and molecular data, all life on Earth is related by descent and could be traced back to some first life form. And the first person to articulate this idea was, in fact, Charles Darwin. He was the first. Not only did he have a hypothesis about natural selection and how life could have evolved, he also had a hypothesis that all life on Earth was related and could be traced back to some single cell that he described as perhaps coming about in some warm little pond somewhere. And that has turned out to be, we can back that up with a lot of data. We do have a lot of data to say that the life on Earth that's alive today is all related by descent. And we'll talk about the three most popular right now competing hypotheses about the origin of life: the RNA world hypothesis, the metabolism first hypothesis, and the protocells or compartmentalization first hypothesis. So first of all, to go from having a inorganic Earth to having complex organic molecules form, that's an uphill reaction. So energy would be required. And life on Earth right now, almost 100% of living things on Earth are getting their energy directly from the sun, if they're photosynthetic, or indirectly from the sun, like us, because we eat photosynthetic things, or we eat things that ate things that were photosynthetic. So all of 100% of human energy comes from the sun, ultimately. There are a small group of organisms, though, alive today that use instead what you might think of as geothermal energy from chemicals that are leaking out of volcanic vents on the bottom of the ocean. We'll talk more about those because those are important for this story. We do know through experiments, and we'll talk about some of these, that amino acids and nucleotides and other complex organic molecules can form spontaneously in the right environment, specifically a reducing environment, meaning that there's no oxygen present. And we've also found amino acids on meteorites from outer space. So that tells us that amino acids especially can form easily in an environment without any life forms present. So what was life like, or what was the environment like on the early Earth? The early Earth had no oxygen in the atmosphere. It had a bigger variety of gases in the atmosphere than we have today. There was a lot of hydrogen gas and helium, water vapor, carbon dioxide, sulfur, chlorine, nitrogen, ammonia, and methane were present in the atmosphere. And there's two ways that we know what was in the atmosphere of the early Earth, because of course no people were around to document it. One way is by looking at the other planets in our solar system, which formed by similar means, like Venus and Mars and the other close by planets, and asking what's in their atmospheres. What is in the atmosphere of a lifeless planet? That's one way. The other way is by traces in the oldest rocks on Earth, which there are rocks that have been found that are almost 4 billion years old. Although with the way the crust is constantly being remelted and recycled, finding rocks that old is rare, and there aren't very many places where you can find rocks that old. Today our atmosphere is very different. Today the atmosphere is almost completely made up of nitrogen and oxygen, about 20% oxygen and about 78% nitrogen. And all of that oxygen and nitrogen is coming from living things. So soil bacteria and ocean bacteria are releasing nitrogen gas, and photosynthetic organisms are releasing oxygen. What happened to all the rest of the stuff? Well, a lot of it is gassed off into outer space. We don't have helium and hydrogen gas in our atmosphere anymore because those are very light. They would have worked their way to the upper edges of the atmosphere, and we're constantly losing atmosphere. We lose tons of atmospheric material to outer space every day. So our atmosphere is constantly gassing off, and without the action of living organisms, we would be more like Mars with hardly any atmosphere. Mars has an atmosphere that's mostly composed of carbon dioxide, and it's only about 2% as dense as the atmosphere of the Earth. Venus has a denser atmosphere because it's so hot that gases are continuing to leak out of the rocks and be baked out of the rocks on the surface. But Venus as well has no life, and it's constantly losing its atmosphere to outer space. So prokaryotes are still doing it, but it was the first living things that completely changed the atmosphere of the Earth and gave us the atmosphere with oxygen that made it possible for eukaryotic and more complex life forms to even evolve. That first oxygen in the atmosphere was originally produced by bacteria, and we'll talk about that. So the very first free oxygen in the atmosphere was about a little over 1 billion years after the first life forms appeared. So the very first cells in the fossil record are about 3.5 billion years old, and about 2.5 billion years ago. So about 1 billion years after the origin of life, traces of the fact that oxygen was now free in the atmosphere begin to appear. It was dissolved in the ocean, causing the precipitation of iron oxide or rust, and that created huge deposits that are still preserved in rocks that we can find today from that age. So this is known as the Great Oxidation Event, and it was probably due to a type of bacteria called cyanobacteria, which are going to be really important for our story later as well for the evolution of eukaryotes. Cyanobacteria first evolved about that time, about 2.5 billion years ago. And although other bacteria before that were photosynthetic, cyanobacteria evolved a new kind of photosynthesis that produced oxygen as a byproduct. And it's the same kind of photosynthesis that plants use today. So plants consume carbon dioxide, they turn the carbon dioxide into sugars and proteins and fats, and they release oxygen from water as a byproduct. And cyanobacteria evolved this type of photosynthesis first. So this was a massive change on the planet, because before that, before cyanobacteria started pumping oxygen into the ocean and then ultimately it ended up in the atmosphere, the organisms on Earth that were alive had never been exposed to oxygen, and oxygen is very reactive and toxic to these sorts of organisms. So there was probably a big die-off at the time of organisms that were sensitive to oxygen. So before that, though, how did we get to those first cells? So one of the hypotheses that has been talked about for around 100 years now is the primordial soup hypothesis. And the primordial soup hypothesis says that, well, we had this warm ocean, there was a lot of volcanic activity, carbon dioxide and nitrogen-containing compounds are being released from volcanoes under this shallow sea, and those compounds then could be released into the atmosphere, where they could be exposed to ultraviolet light, which could cause things to bond together. There's our energy to bond things together. And then they would rain back down into the ocean, and the cycle would continue, and eventually you get more and more complex molecules, and then at some point eventually cells. That's the primordial soup hypothesis. And it was tested experimentally, and this experiment is still going on. A graduate student named Stanley Miller, who was in this guy's lab, Urey, I can't remember Urey's first name. His graduate research project was to test the primordial soup hypothesis. So he created a closed system, and you can see where's my pointer here? He created a closed system where he could keep the oxygen out and keep out living organisms that are alive today. Because, of course, if it was open to the atmosphere, bacteria would get in there and oxygen would get in there. And he was trying to recreate conditions of the early Earth. So at first, he just heated water that contained methane and ammonia, and he had some hydrogen gas in there. And he was letting it bubble around and then recondense and fall back down. And then he was trying to mimic rain. But it turned out, and nothing happened. It turned out that that was not enough energy. So he added sparks to give, he was thinking this was mimicking lightning. Because there, if there was water and thunderstorms, there would have been lightning. And immediately, as soon as he added the electrodes and added the sparking, that higher energy, immediately within a day he found organic material in this chamber. And as he allowed the experiment to go on, more and more complex organic molecules continue to form, and it's still going on today. This experiment is still going on. And they have found many different categories of molecules, although nothing alive has come out of the experiment. There's a video, the nice little summary video that I will post on our page. So it's too long to show during this lecture portion, but I'll give you a link to it so that you can watch it. It's a nice little 6-minute animation summary of this Miller-Urey experiment. So this was really a cool experiment. It was very big news at the time when he first reported his results in the 1950s. And although Stanley Miller is passed away now, and so has Urey's been dead way long before, the experiment continues. Another test of the hypothesis was done by, as another graduate research project by this woman, Tracy Lincoln, and she was specifically testing the RNA world hypothesis. So the RNA world hypothesis says that, well, maybe the first thing that was replicating wasn't a cell, but was just some kind of nucleic acid. And RNA, we know now, since the 1990s, that single-stranded RNA can act as an enzyme. It can not only code genetic information, it can fold up into a three-dimensional shape like a protein and act like an enzyme. And so she created synthetic RNAs that had enzymatic activities, mixed them together, allowed them to do their thing, and she showed that mutations would accumulate in these RNAs, and they would actually work faster. So that showed that evolution would take over. Once you had a molecule that could self-replicate, evolution would start to occur, natural selection would start to occur. But she had to feed the experiment the raw materials. The RNAs were not replicating de novo, with no RNA as a raw material. She was giving them pieces of RNA, and they were rearranging the pieces essentially to make a copy of themselves. So it was very cool, very supportive of the RNA world hypothesis, but not definitive proof of the hypothesis. So even though RNA seems like a really likely suspect for the first replicating molecule because it can act as genetic information and an enzyme, no RNA has been shown to completely synthesize itself. So even recreating RNAs of any desired sequence in the lab, which we can synthesize small RNAs of any sequence, scientists haven't been able to create one that can completely replicate itself. So that, unfortunately, does not support the RNA world hypothesis. So another hypothesis has been proposed, and that is that perhaps we're too focused on the nucleic acids and replication, and that perhaps the first thing a living thing had was metabolism and not the ability to replicate itself. Because that's another thing that all living things today have in common, is certain elements of the cellular metabolism are in common. So there are various metabolic cycles, especially metabolic cycles that involve acetyl-CoA, which we'll talk about when we talk about respiration, that all cells have in common. Another thing that all cells have in common is using proton gradients to generate ATP molecules. You might have heard of ATP. We talked about it briefly in the biochemistry section. ATP is an energy storage molecule that every cell on Earth uses. And cells make ATP from ADP, which is a less energetic version of ATP, using a proton gradient or a hydrogen ion gradient. So remember, hydrogen has just a single proton in its nucleus. So a hydrogen ion, an H+, is just a single proton because it only had one electron. We take that away to make it positive, and now it's just a proton. So when we say proton, proton is also in biological chemistry a synonym for hydrogen ions. So using a proton gradient, going from a high concentration of protons on one side of the membrane to a low concentration of protons on the other side of the membrane, our cells use this, bacteria use this, every cell uses this to make ATP. So that's another thing that cells have in common. So maybe that was first. Maybe making ATP with a proton gradient, or maybe one of these other metabolic cycles that involves acetyl-CoA was the first thing, not RNA. And where would you find a proton gradient loose in the ocean? Well, it turns out that although volcanic vents at the bottom of the ocean tend to be acidic, almost all of them are acidic because they're producing carbon dioxide. And remember, carbon dioxide is a weak acid, or it forms a weak acid. It forms carbonic acid when you dissolve it in water. So most volcanic vents, because they have carbon dioxide being produced, they're going to be acidic. But some alkaline hydrothermal vents at the bottom of the ocean have been found in the Atlantic Ocean, and these tend to be cooler as well, and they tend to be at temperatures that are much more compatible with biological molecules. They tend to be, the acidic hydrothermal vents tend to be super hot. Often they're above 100 degrees Celsius near the vent. But these alkaline vents are much cooler. This is a picture of them over here. They're still hot, but too hot for people, but they tend to be more like 75 degrees Celsius, which would be much more compatible with the formation of some kind of protocells. So, and then here's our third hypothesis. What formed first? Well, maybe it was some kind of protocell, because it's a watery environment. Fats tend to stick together. If you had anything resembling a phospholipid, they would tend to clump together in spheres naturally. And all you would need is to maybe have some kind of RNA inside. And this picture here is actually of one of these protocells created experimentally in the lab. Unfortunately, though, when the protocells that have been lab created, the RNAs and other things that they put inside don't do anything. So, but those are our three competing hypotheses right now. RNA first, some kind of replicating RNA molecule, which is called the RNA world hypothesis. Some kind of metabolism first, either some kind of a metabolic cycle or something that's creating ATP with a proton gradient first, or maybe little protocells formed first with some kind of a lipid molecule that separated inside from outside, compartmentalization first. So those are our hypotheses right now, but we don't have solid evidence of any of them. There seems to be a lot more laboratory experiments with the RNA stuff, but that doesn't mean that that's the one that's going to win out in the end. So early Earth, we have our base molecules here, which can be used to make all of our complex molecules. So you'll recognize here's our four big categories of biological molecules: amino acids, nucleotides, carbohydrates, and lipids. And the Miller-Urey experiment showed that you can get all of these spontaneously if you have conditions right, if you have no oxygen and if you have an energetic enough source of energy, like sparks versus just heat isn't enough energy. You need something higher energy than that. Going from these building blocks to polymers, either proteins or complex RNA molecules or DNA molecules or polysaccharides, this step has sort of been demonstrated in the lab, that it has been demonstrated that you can get nucleotides to link together to form long strands if you provide them with something to bind to, like clay. But then the question is, how do you go from RNA to DNA? All living things today use DNA as their genetic material. How did we get from that? If there was an RNA, was the first replicating something, how did we go from that to having DNA be the genetic material? Was compartmentalization first? The little lipid spheres engulf something, and that ended up being the first protocell. So there are people going at this problem from the other direction and trying to create a cell completely from scratch by synthesizing phospholipids, synthesizing synthetic RNAs and DNAs, and putting them together in their synthetic protocells and trying to see what is the minimum number of ingredients that you need in order to get something that sort of resembles a replicating cell. And so far they have gotten their little completely artificial cells to take up things from the surrounding, and to even, when they get very large, to split. But that's about as far as those experiments have gotten as well. So lots of people working on this problem, but not a lot of definitive answers yet. Lots of intriguing data, but we may never know for sure exactly what happened to give us the first cells. What we do know more about, though, is what that first cell was like. And this is called, we call this first cell that's the ancestor of all the cells that are currently on Earth, the last universal common ancestor. And this may not have been the only living thing at the time. There may have been thousands or millions of variously dividing different things. None of them survive to this day. What we have today are descendants of these first cells that we call LUCA, for last universal common ancestor. And what LUCA was like has been deduced by studying the DNA of all of the prokaryotes that are alive, or a sampling of all of the prokaryotes that are alive. And scientists ask the question, okay, because prokaryotes were the first living things. For the first billion years of life on Earth, there was only prokaryotes. So those would be the organisms to study. So if they compare all the prokaryotes that are alive today, compare their DNA and ask, okay, what genes are in common to all of them? Because if every living species of prokaryote today has particular genes, probably the ancestor also had those genes. So that they asked that, and they got about 350 genes out of a typical prokaryote has 3,000 to 4,000 genes. So they found about 350 genes that are common to all prokaryotes. And that gives them an idea of what kind of a lifestyle this first prokaryote had. So this first prokaryote, the LUCA, was alive around 3.5 to 3.8 billion years ago. The earliest fossils that we have that definitively show of cells about the size of bacteria today are a little bit younger than that. So we don't have fossils of these first cells, and that would seem unlikely that we would. But we do have fossils from a few million years later that show that they were about the size of bacteria. So because every prokaryote alive today has a circular DNA chromosome, probably LUCA did too. And these 350 genes that are in common indicate that this first cells probably lived around deep ocean volcanic vents and were anaerobic, meaning that they did not use oxygen as part of their metabolism, and they weren't photosynthetic. Instead, they used hydrogen sulfide for energy. And there are bacteria today that still do this. This is a picture of one of those volcanic vents. These are the acidic ones, the more common ones. They're found all over the world in various places, and these are called black smokers, obviously, because that's what it looks like. They're extremely hot. The water around them is very hot. They're very deep in the ocean often, and it's a very high pressure environment as well. Yet there are bacteria that live there and live off of the hydrogen sulfide. They split hydrogen sulfide for energy. There's a lot of energy in the bonds of hydrogen sulfide, and that's what they use to make ATP. And they use carbon dioxide as their carbon source, just like for photosynthesis. And they could also fix nitrogen. They could take nitrogen and use it and turn it into a form that they can use to make proteins with. And they were most like a simple bacteria. So the earliest fossils that we have, which weren't LUCA, but descendants, the earliest descendants of LUCA that we have fossils of, they were about the size of bacteria today, a few microns in diameter. So this is what we think the first true cells were like. So what happened to all the other stuff that was reproducing in the oceans at the time? They all went extinct. Some people hypothesize that maybe some of those early life forms, other types of life forms, maybe that's where some of the viruses came from that we still have around today. There are many viruses that attack bacteria today, and perhaps some of those viruses that attack bacteria are remnants of other life forms that were around at the time. But they're not cells, and we don't consider viruses to be alive. So here's the history, the entire history of our planet, put on what you could think of as like a clock. So the entire span of time here is a little over 4.5 billion years. That's how old our planet is. And at some point, the first cells appeared about 3.5 billion years ago. Those were the first cells, and the first cells that we have fossils of, they look just like bacteria that are alive today. And then there was a very long period of time where there was nothing but prokaryotes. Remember, prokaryotes don't have a nucleus. They're the bacteria in the archaea. And there's still a lot of them around today. So there's a very long period here, over 1 billion years, almost 2 billion years, where there's nothing but prokaryotes. They're the only life on Earth. And here's where the cyanobacteria first appear and start producing oxygen. So very low oxygen. Now the oxygen in the atmosphere here never got above around 2%. Right now it's at 20%. So pretty low, but enough oxygen was around in the ocean to allow eukaryotes to appear. So eukaryotes are oxygen dependent. Our mitochondria use oxygen to make ATP. And we'll talk more about that later in one of the later chapters of this, or later videos for this chapter. So if single-celled eukaryotes appear, and then there's a very long time again here. So we have 1.5 billion years where everything is single-celled. We have prokaryotes and single-celled eukaryotes. How do we know they're eukaryotes if they're single-celled? Well, they're much, much larger. So suddenly in the fossil record, these cells appear that are 100 times the size of bacteria. Those are the first eukaryotes. And then eventually some multicellular eukaryotes appear in the fossil record, and these are algae kind of things, leafy looking things that today we would consider protists. And then a very long time later, the first definitive fossil animals appear. You can see we're getting very close to now. It's very late. So this long, long period where there's nothing but single cells, even paleontologists call this the boring part of the fossil record. But I think it's really good to see it on in this clock kind of format, because it really shows how brief animal presence is on the planet. And we think of it as being a long time, because when we think of fossils, we think of dinosaur fossils and fish fossils and, you know, megalodon and all these other things. That was very recent in the history of the Earth. Okay, so finally we have, life is all in the ocean at this point. And then very recently, only a little more than 500 million years ago, we finally get the land being colonized. And it's first colonized by fungi, bacteria, and then plants, and then finally animals colonize the land. And if we look at here, the names of these periods here, so the Mesozoic, this little orangish one here, this is the age of the dinosaurs. Before this, the Paleozoic, sometimes this is called the age of fishes, because this was when land animals first appeared, which the first land animals were arthropods, insects, and then later amphibians and reptiles. They appear first during the Paleozoic. Mesozoic is the age of dinosaurs, so dinosaur extinction is right here at the boundary between the Mesozoic and the Cenozoic. That was 65 million years ago when the dinosaurs went extinct. And then finally we have the appearance of primates shortly after that, about 60 million years ago, appearance of the first primates, appearance of the first hominin ancestor about 8 million years ago, the appearance of our species only about 300,000 years ago. So very recently in the history of Earth. So, so we're going to talk a little bit later about the origin of eukaryotes as well. So what about life on other planets? We know a little bit about life here, and you know, people have been speculating and wondering about life on other planets for at least a couple hundred years, since we knew that the planets were planets and not some kind of star or something. So far, we have zero evidence of life anywhere else, either on planets in our solar system or outside of our solar system. There's zero evidence of life on Mars. There was somebody a geologist, who reported many years ago, maybe it was as long ago as 25 years ago, that he thought he had found in a Mars rock fossil cells. But not being a biologist, he didn't realize that the size of the thing that he was looking at was so small that it was highly unlikely to be a cell. It was about a 1/100th the diameter of the smallest known cell today. And it turned out that other geologists proved that what he found was not due to anything alive, but could be produced completely naturally without any living things. So he was just seeing essentially tiny bubbles in the rocks. People have wondered about the planet Europa, or the moon Europa rather, because it's covered with water. It would be really hard for photosynthesis to be occurring there. Perhaps there are hydrothermal vents under the surface of this ice where something could have evolved that doesn't depend on sunlight. There is liquid water under the surface, but so far there's a lot of speculation, but we have absolutely zero evidence that any life ever evolved anywhere else. But we haven't really explored outside of our solar system yet. Mars used to look a lot more like the early Earth. Mars used to have a dense atmosphere. It used to have liquid water on the surface, just like the early Earth did, which led some people to speculate that perhaps life arose there and then went extinct. So far, we don't have any evidence of that, but you can look at Mars and see what Earth would look like if life had never evolved here, because Mars has lost almost all of its atmosphere to outer space. That would have happened to Earth as well. And in fact, we're constantly losing our atmosphere. The reason that we have the dense atmosphere that we have today is entirely because of living things continuously producing the gases that are found in our atmosphere. No life yet anywhere else. So how we classify living things here has changed over time. Linnaeus, who we talked about previously, who came up with the classification system that we use today. He's the one who came up with the genus and species, the two-word naming system that we still use today. He came up with the kingdom, phylum, class, order, family, genus, species system that we still use today. But he only had two kingdoms. Everything was a plant or an animal. So if it moved, it was an animal. If it was photosynthetic, it was a plant. If it couldn't move, it probably was also a plant. So fungi were plants in Linnaeus's classification system. And this kind of fell apart when you got to the single-celled organisms, because there are single-celled organisms that are both photosynthetic and swim. And there are bacteria that are photosynthetic, which he would put in plants. And then there are bacteria that are heterotrophs, which he would put under animals. So it broke down at that level. So about 150 years later, this guy Whittaker, who was around the turn of the century, the turn of the 20th century, so 120 years ago, he came up with a new kingdom system that got rid of that problem. He took all the protists and he put them in one kingdom called Monera, for single-celled, from the Greek root mono, for one. And then he created the other kingdoms, three of which are still valid today: protists, fungi, plants, and animals. It's the protist kingdom that's no longer valid today. So Monera has not been used for a long time, not since the 80s. So if you find a source, a blog, or a Wikipedia page or something that mentions the word Monera as being a valid kingdom for bacteria to be in, you know it's 40 years out of date. So what happened? Well, in the 1970s, this guy at the University of Illinois down in Champaign, Carl Woese, he was sequencing genes, and this was really the beginning of what would be a DNA revolution. Being able to look and compare the sequences of different organisms, Carl Woese came up with that idea. So he was sequencing a specific gene, a gene that's present in hundreds or thousands of copies in every cell. the ribosomal RNA genes. So these are genes in the DNA, in the chromosome of the organism, that code for an RNA that's used to transcribe protein, or translate protein, I should say. And he discovered that organisms that are closely related have almost identical ribosomal RNA genes, and if they're more distantly related, there are more differences between the genes. So he realized, hey, I can use this gene to compare how related different organisms are. So he was comparing bacteria, and he compared them to eukaryotic genes and said, oh yeah, look, bacteria and eukaryotes are way different. They're totally different kinds of ribosomal RNA genes. And then he found one bacteria that he was studying in his lab, a methanogen, so produce methane as a byproduct. And he sequenced its ribosomal RNA gene, and it turned out to be just as different from bacteria as eukaryotes are. And he was like, oh, it looks like a bacteria, it's a prokaryote, but the gene indicates that it's very distantly related from all the other bacteria. In fact, its ribosomal RNA gene is more like a eukaryote, it's more like us. And that really set off a whole sequence of research that led to the domain system that we have today. So Carl Woese eventually said, okay, we need something above kingdom level. There are three big domains, or three big splits in the tree of life: bacteria, archaea, which he named, and the eukaryotes. And he named the archaea that because at the time he thought that he had discovered the organisms that were closest to LUCA. So archae, that prefix means first. So he named the archaea that because he thought these were the closest to the first cells. That turns out not to be the case, but the name sticks because the person who discovers something gets to name it. So we now think that the first cells, the LUCA cells, were actually more like bacteria, and further sequencing has shown that. So archaea are actually the ancestors of the eukaryotes, and we'll talk more about that in the next video. But that's where we got this domain system that we have today from Carl Woese. It's like the next level up, and it represents an earlier branching in the tree of life. So here's our tree of life today. Here's LUCA down here, this thing that says common ancestor. That's the root of the tree. That would be those very first cells that had a circular DNA chromosome and were living around hydrothermal vents. There was a very early split, and we don't know exactly when this split happened because we can't see it in the fossil record, because archaea and bacteria look the same in the fossil record. So we can't tell exactly when this split happened except through DNA sequencing. And we know that it was very early, maybe as early as 3 billion years ago. Bacteria and the archaea split, and they've been separating ever since. And then later, about another billion years later, the first eukaryotes appeared, and they were a modified archaea species. And we'll talk more about that in the next video.